Welcome to Unit 2 in AP Biology, and today we will just recap over the cell organelles and their functions, the compartmentalization inside of the cell, as well as the endosymbiont theory that is supported by many evidences. So let's start. I made a PowerPoint for easier to understand, and I will write on it too. So the first one and the second section in that unit is cell structures, components, and their functions. We distinguish two types of cells in biology: the prokaryotic cells and the eukaryotic cells. They look similar to each other, although the prokaryote is way smaller than the eukaryotic cell. Now, the similarity for both of them is that they have a cell membrane, both of them, that surrounds the cell. The cell membrane keeps the internal structure and components inside from the outside. Same in here. This is the fundamental importance for the cells, for living organisms to keep their homeostasis in here compared to the outside. Homeostasis, same internal environment for them. What does it mean? Not necessarily temperature, but the ionic concentration, solute concentrations, and something like that. Both of them, the prokaryotes and the eukaryotes, have ribosomes. They are the sites for protein synthesis. Protein, last time we just discussed, they are the working horse for the cells. Cell communication, signaling to each other, receiving signals, transmembrane channels all around in the membrane. Enzymes, they are all made out of proteins, as well as structural parts of the cell. The next one is the cytoplasm, the gooey-ish material inside of the cells. And the last one that I would like to point out, this and that, and I would just say DNA. So this is the same for both of them. On the other hand, the DNA for the prokaryotes is circular. For the eukaryotes is linear in structure. So these are kind of like that. And for the prokaryote, it's circular in shape. It will be important later on. Now, it's really separated from the cytoplasm in prokaryote in the nucleoid region, but it's not circled or enclosed in a membrane-bound shape. The eukaryotes, what makes them eukaryotic organisms, that these organelles, many of them, are membrane bound. Membrane encloses them. So what they do inside of their organelles, each organelle is doing something inside of it, will not get affected by the surrounding cytoplasm. So they can be more efficient on doing something, and they can do many things together at the same time. I think that's all for that. Please refer to the apply assignment about the function of each organelle in the eukaryotic cell. Here is that. Now, the next topic is the cell size. You have to be able to calculate the surface area and the cell size for different shapes. You have the equation sheet on AP Biology, but you have to practice how to do that. Now, cell size and the size matters. Why are cells so small in our body? Why can't I have 3 cells in my arm? 1, 2, and 3. Because the surface area over volume ratio matters. As the cell is small and gets bigger, the surface area of that small cell is small. The surface area of that big cell is big. The volume is small, the volume is big. But the point is the ratio per unit volume. So the ratio, the surface area for that volume is way bigger compared to the surface area for that huge volume. Why is that? Because the volume is increasing kind of by 1,000 times, so it's 10 to the third time, and the surface area is just only squared. Anyway, so to have a large surface area volume ratio is beneficial for diffusion, absorption through the cell membranes. Now, think about the lung. The lung is made up of alveoli. These are grape-like structures, and these alveoli are increasing the inner surface area for the lung. On the outside of the lung, we have vessels, capillaries, a little bit of blue, a little bit of red. Okay, so these are the capillaries surrounded where the gas exchange, the oxygen goes out from the alveoli into the blood vessels, and and the carbon dioxide from the blood vessel goes into the alveoli to refresh the blood for the organism. The bigger the surface area, the faster this exchange can happen for the organism because of the surface area is bigger. Same concept on the absorption. The small intestine is the place to absorb the nutrients, what got digested in our digestive tract. Not only we have folds inside of the small intestine, but also the cells on the folds have microvilli, tiny areas that increase the cell surface area for absorption. Same concept for the root hair. The root hair has tiny extensions, the cell cytoplasmic extensions to increase the surface area for nutrient absorption for the plants. Think about the nitrogen, they have to get it from the soil, the fixed nitrogen and the phosphate. And also the water for their photosynthetic activity. So this is also larger the surface area over volume ratio, the better for the plants. Here is the elephant. I want to just show in here the big ears acts like a huge surface area over volume ratio for the elephant. Why is that so important? To get rid of the excessive heat when the elephant is too hot because of the climate or the area where it lives. Now, sometimes we aim for smaller surface area over volume. When is that? At the Arctic area, for example, the polar bear, it's a huge animal. Trust me, it is. And the surface area relative to the volume is smaller for the polar. Why is that super important? Not to lose heat during cold season. That is the majority of the year. So they can preserve their heat inside of their bodies. So surface area over volume ratio that is small, sometimes it is useful. Think about when you are cold, you want to just make yourself smaller or just huddle all around because you want to keep the heat inside. That's it for this. The next one, what I would like to talk to you about is the compartmentalization. Now, compartmentalization means that each section in the cell, in eukaryotic cells now, the sections and organelles are enwrapped in membranes to separate their working environment from the cell itself. For example, the lysosomes. The lysosome is, they are here all around, and when the cell eats or engulfs something through endocytosis, that will be our next week material, the food needs to be broken down. What breaks down is the lysosome contains hydrolytic enzymes, hydrolysis, splitting up the big macromolecule. Think back last week material, and going to be chopped up. In here, the lysosome fuses with the food vacuole and breaks up the material inside. And after, what it's going to be used, what it's not. So it's exocytosis. By exocytosis, the cell gets rid of the waste. Now, also, we will learn later on about apoptosis. Apoptosis is when the cell kills itself. How will that happen? It will get a message, the lysosome, to break down its membrane. All the enzymes, the hydrolytic enzymes, are going to get leaked out into the cell, so to destroy itself. Sometimes we need that we need that the cell would destroy itself. For example, when the cell cannot repair its DNA mistake, and it wanna just turn into a cancerous cell, normal signaling cells can make themselves go through cell dying, apoptosis. That was this. Another one is the mitochondrion. We have two membranes. Later on, next will be super important. We have the outside membrane and the inside membrane. So check that, how convoluted the inside membrane is compared to the outside membrane. Why is that? Because the inside membrane contains proteins in here, and they are the electron transport chain proteins. We will learn that in the energy unit through cellular for the cellular respiration. And they have a membrane embedded enzyme that is called the ATP synthase. It makes ATP. So bigger the surface area, more ETC proteins and ATP synthase proteins can be embedded in the membrane, so the efficiency will be higher for the mitochondria to produce ATP. Same with the chloroplast. They have two membranes and the thylakoids. The thylakoids again increasing the surface area because inside they are hollow, and the pH, and also for in here in the mitochondria, the pH in here is different than inside of the thylakoid. The pH outside in the matrix area is different. In the intermembrane space is different than inside of the matrix. We will learn about that. That will be one of the key importance why the mitochondrion can produce ATP, what drives the mechanism. But hold on to it. This will be in a couple of weeks later. So we did this, good. And the last one, what I would like to talk about, that's the endosymbiont theory. In science, the theory is something that is really well supported by evidences that it is true. It's not kind of, I have a theory that today it will be a nice weather. Okay, fine, but it's more than the English theory, actually. So, supportive facts for that. What does it mean? Endosymbiont, endosymbiosis or symbiosis theory that we had long time ago, couple of 100 million or even more, like 2 something billion years ago, we had a prokaryotic cell. We had the nucleoid in here with the DNA and the cell membrane, nothing else. Now, somehow the cell membrane started to vaginate, enfold inside of the cytoplasm, and later on it made the nuclear membrane around the genetic material, and the endoplasmic reticulum started to form, getting off of the cell membrane, as well as the Golgi body. Now, this is going to turn into a eukaryotic cell because it has a nucleus, it has a nuclear membrane around the hereditary material. Along the way, there came a bacteria that was able to be more effective producing energy with the help of oxygen compared to the cell that used glycolysis process to make very, very limited amount of ATP. So the mitochondrion got fused in or was eaten by the cell, but the cell did not break down the mitochondrion. They started to live together in the cell. So this will gave rise to the cell that they are heterotroph cells. On the other hand, there was another endocytosis when the cell not only had the mitochondrion inside, but ate a cyanobacterium. The cyanobacterium is a bacterium that can do photosynthesis. So it can make its own food, so it's an autotroph organism. Bacteria did not get eaten, so they live happily inside of the eukaryotic cells, and this would give rise to the photosynthetic eukaryotic organisms, like plants or green algae. Now, this is the story. What supports that story that it is actually true? Now, so the mitochondrion and the chloroplast, they both have circular DNA. We mentioned that at the beginning, only prokaryotes have circular DNA. Eukaryotes, linear DNA. They have that. Number two, they have bacterial / prokaryotic ribosomes. Ribosomes, I mentioned at the beginning, prokaryotes and eukaryotes have ribosomes. They are almost kind of the same, but they are not exactly the same. Prokaryotes have a little bit of different ribosomes compared to the eukaryotes. And the mitochondrion and the chloroplast have prokaryotic ribosome structures inside of their bodies. And number 3 is they divide by binary fission. Reproduction, repro by binary fission. Now, and independent from the cell, they can make more of themselves by binary fission instead of mitosis. And we will discuss that when we hit the cell reproduction. I think that's all for this week. And if you have any questions, just send me an email and have a good day. Bye.